Invisibly, they hide beneath our feet. For centuries, we didn't even know they existed. But now, they shape our future. They open up amazing possibilities, but at the same time threaten to destroy our environment.
If you look at the periodic table, there's a couple of rows that appear to be kind of kicked out. They don't quite fit in there, and they're down at the bottom, and people tend to ignore them.
The reason why we started focusing on them is that they have a number of very exciting technological properties.
And now for something completely different.
I started a show full of fun and unintentional things. DaHoShow, streaming on YouTube for thee. DaHoShow, the courts have compelled me to warn you, it's me you will see. Streaming and gaming and talking and fussing. DaHoShow, it's free. Oh, say, I started a show full of fun and unintentional things. DaHoShow, streaming on YouTube for thee. DaHoShow, the courts have compelled me to warn you, it's me you will see. Streaming and gaming and talking and fussing. DaHoShow, it's free.
Once isolated, they have properties that allow a myriad of modern technologies.
Smartphones and tablet computers, hybrid cars and electric vehicles. All our modern gadgets contain rare earths. Baotou, China, the world's largest rare earth mine. 97% of the world's supply of rare earths is mined in China.
Apart from promethium, which is very scarce, all the other rare earth elements can be found in China. And when you talk about rare earths at the moment, you automatically talk about the resources in China.
The open-cast pit at Baotou is in fact an iron ore mine. It started production in 1927. For decades, rare earths were considered a waste product, until prices began to rise on the world market.
A couple of decades ago, the Chinese discovered that the material which was above iron ore in a place in Inner Mongolia was in fact rare earth, and that they could, from this material that had been dug up and cast aside, they could separate rare earths. And they started producing unseparated rare earth oxides.
So has China become the world market leader only by chance? The head of the Chinese Society for Rare Earths thinks differently.
In the 50s, China began the exploration of rare earths. In the 60s, we built our own processing system and began mining operations. In the 80s and 90s, we started mass production.
No doubt. By now, we all depend on China. Rare earths mined and refined in China with dirty and outdated methods are bought by high-tech companies around the world. to produce smartphones, electric cars, and wind turbines. Matthias Buchert from the Institute for Applied Ecology in Darmstadt knows how difficult it is to produce rare earths.
You have to bear in mind that rare earths never occur on their own, but always in groups. They're chemically very similar. To separate them, you need very sophisticated processes that need lots of chemicals and energy. Only then you end up with the pure elements or metals.
Bremen, Germany. Rare earths are already in short supply, and they're expensive. Global consumption has doubled during the past 20 years and is still rising steeply. Therefore, researchers are looking for new deposits around the world. Andrea Koscinski from the Institute of Geochemistry investigates rare earth deposits in the Pacific Ocean. She's particularly interested in these strange nodules, which are full of useful raw materials.
There's a demand for raw materials, and there are deposits on land. But many natural deposits are in remote, sensitive ecosystems. In the rainforest, for example, or in coastal areas, or they're mined in countries where people are working under inhuman conditions. All these things could be avoided with deep-sea mining.
Hawaii. In the port of Honolulu, a research ship sets sail. On board, scientists searching for rare earths.
We've known for some time that manganese nodules and manganese crusts on the ocean floor have a relatively high metal content, especially nickel, cobalt and zinc. But due to new technologies like green energy generation and so on, the demand for rare earths is rising. And we're now more interested in these elements because of their economic value.
Diving robots are scouring the seabed for manganese nodules. Since 2006, the German Federal Institute for Geosciences and Natural Resources has been exploring 2 sites in the Pacific Ocean. A video camera shows images of the ocean floor. But is it really so easy to simply pick up natural resources from the seabed? Environmentalists are worried about the destruction of the ocean floor.
There are quite complex life forms down there, and processes in the deep sea are very, very slow. Everything grows and regenerates slowly. So if we interfere with such a system, it's difficult to predict how long it will take before it regenerates.
Scientists hope to find millions of tons of rare earths in the Pacific, enough to make us independent of China's supplies.
This is an area of research we've only just started to explore, but we already feel that it'll have a much greater importance in the future. This is one of our big challenges. If we want to harvest these nodules, how do we get the raw materials out of them? For example, rare earths.
Andrea Koscinski wants to find out what's hidden inside these manganese nodules and how to get it out.
Genau, es sieht gleichmäßiger aus. Ja. Der Kern ist auch wirklich in der Mitte. Ja, sieht man eigentlich sehr schön.
You can see here that both these manganese nodules have a core. They need some kind of seed to start growing. This seed, like this little stone here in the middle, just sits somewhere on the seabed, in the sediment, at a depth of 4,000 or 5,000 meters. It could be a fragment of a rock, or a fish tooth. Now, deep down there, the seed is chemical processes start depositing this black material, manganese and iron oxide. we also look at the entire spectrum of elements to get an idea of the makeup. If you want to describe rare earth, you have to get backwards back into the elements. Notice the lots of rare earth minerals that you pick out with the It's complicated. You have to understand first because the black stuff is very
Analyses show that manganese nodules contain high amounts of rare earths. That are urgently needed for how green is our future if we have to destroy pristine ecosystems?
Consider that there are many countries in the world that still have somewhat low building infrastructure for an adequate standard of living. If these countries are to use green technologies to get there, then they need new resources, like, for example, rare earth.
To build more wind turbines and solar energy plants around the world, we have to exploit new rare earth deposits. The difficulty is finding them in the first place.
For metals like iron, chromium, zinc, lead, copper, and so on, there are very specific deposits. So we have zinc deposits, we have lead deposits, well-defined structures from which we can easily obtain metals. This is not the case with rare earths.
Right in the middle of the geologists follow a hot wind. During GDR times, scientists were looking for uranium, but instead they found rare earths. Nobody was interested in these strange elements back then, so the mine shaft was closed. But now a mining company is test drilling again, hoping to tap into the largest deposit of rare earths in Europe.
If you're looking for investors and you tell them we have some GDR documents, then most investors will say, well, yes, but they were written during the socialist GDR period, so we don't really trust them. This means we need to reconfirm the old data.
Geologists drilled deep into the rock. 4.4 million tons of ore is supposed to be here. A blessing for the German high-tech industry, which is completely dependent on rare earth imports from abroad. But how to excavate the treasure? A huge open-pit mine in the middle of Germany is not an option.
This rare earth deposit has a diameter of 130 meters and a depth of 1,000 meters. This would make a gigantic hole. We can't do this. Our best option is to build an underground mine shaft. This means we only need a deep pit with a small tower at the surface and a few buildings around it. The entire mining operation would happen underground.
An expensive, technically complex deep mining shaft for a tiny deposit. Is this economically viable? The data from the test drilling needs to determine the concentration of rare earths in the earth.
Gold sparkles beautifully. Diamonds can also be seen easily. As for rare earths, they're very inconspicuous. The stuff looks like cement. It's grey and nothing sparkles. Nothing catches the eye. We only know from geological analysis that there are rare earths. And in fact, even if water can say, yes, rare earths are in fact present. But we can't see them with the naked eye.
Neodymium makes magnet stronger. Yttrium ensures vivid images on computer screens. Lanthanum makes electric car batteries more efficient. But for a long time, Scientists simply didn't know what to do with this strangely named elements. Sirius was discovered in 1803, but it took more than 100 years until an inventor came up with a bright idea of what to do with it.
Die ersten Anwendungen des Werden waren gewesen der Anwendung.
The first applications for rare earths were essentially limited to the element Sirius.
You got my attention.
There were two applications. One was using it for lighter flints, and the other one was making incandescent lanterns for gas lamps out of it. This was
Great to meet you. How are ya? What can I do for ya? What's on your mind?
Later,
Hi?
applications were found for rare
Think
earths.
that's funny, do
For
you?
example, made the
You're
production
not
of gasoline
amusing.
much easier and cheaper.
That's not funny. Watch it. How are ya?
The price of gasoline would probably be between
My, you're a tall one.
30% higher than what it is right now.
Interesting.
For rare earths.
Making a special
And it's
purchase?
already quite high.
Would you like to purchase something? Hey, are you
Mountain
through?
Pass, California.
Was there something else?
Long before China,
Yes, you're
the
totally
US was the
annoying.
main producer of
Sorry, I
rare
wasn't
earths.
listening. Pleased to meet you. Good day to you. My, you're a tall one.
The simple answer to this is family right now.
Off with you.
The trick is finding deposits of rare earths that are rich and long.
For decades, the mine at Mountain Pass was considered the most important rare earth deposit in the world.
The start of the Mountain Pass district was about 1949 when prospectors found a radioactive signature on some outcropping of rock. They analyzed it and found that it was remarkably rich in rare earth.
The problem with deposits like Mountain Pass is that rare earth elements are usually closely bound to other elements. Many of these contaminants are radioactive. To isolate rare earths, mining companies use sulfuric acid, nitric acid, and hydrochloric acid. This produces highly toxic radioactive effluents. The reason why rare earths do not exist as individual elements in nature lies in the geological formation of the deposits.
Geologists tell us that there were 8 volcanic intrusions which molten carbonatite rock came up through that crack and was deposited layers and built up the whole deposit.
Between 1965 and 1995, the mine at Mountain Pass was the world's largest producer of rare earths. The raw materials were needed for making lighters, gas lamps, color television sets, and for producing gasoline. However, the production processes at Mountain Pass were complicated and prone to failure. In 1998, the worst-case scenario happened. 1 billion liters of radioactive and chemically contaminated leaked from a collecting pond and polluted a nature reserve. Stricter environmental regulations made the mine unprofitable. In 2002, Mountain Pass closed.
Shall I schedule this anyway?
Shortly after the closing of Mountain Pass, a technological revolution happened. Smartphones and flat screens took the world by storm. At the same time, environmentally friendly cars and alternative energy sources became popular. All these modern technologies need rare earths.
We're currently 7 billion people. In 40 years, we'll have an additional 2 to 3 billion. These people need resources, not only drinking water, but also rare earth metals, which are used in mobile phones, cars, or wind turbines. This means that the demand for raw materials will increase tremendously in the future.
Beijing, China. At the same time as the Americans withdraw from the rare earths business, the Chinese take over. The new economic superpower is able to produce raw materials China's boomtowns are skyscrapers and mushrooms, but the domestic demand for consumer goods is rising. For example, China has the largest number of mobile phone users worldwide. As demand for smartphones and flat-panel displays rises, China's production of rare earths increases accordingly. One by one, mines in other countries close down, unable to compete with China. The world is now almost entirely dependent on China's rare earths.
During the last 10 to 15 years, China has taken over not only mining operations, but also the separation and production of refined rare earths. Around 97% of all rare earths are now produced in China. They are in a very dominant position.
But in 2011, China's government the mines reduce their output accordingly. The world gets a feel of how powerful China has become. Because prices shoot up on the world market, energy-saving light bulbs, for example, suddenly get 25% more expensive.
The problem of the reduced exports from China really is one that has been in the time. China reduced the exports for a number of reasons, one of which they have a very rapidly growing internal market, and that they want to manufacture finished goods within their country rather than ship unfinished rare
The reasons for this export restriction. Is it because of environmental problems? Or are the Chinese simply flexing their muscles?
I don't want to rule out that the restriction of rare earth metal exports has something to do with the tightening of environmental regulations. But it almost certainly has also got something to do with China's power over the world market. I'm certain about that.
So far, China's forte has been mining. But a lot of money can be made with processing, which is complicated and difficult. Right now, processing takes place in Japan, Europe, and the US. Many experts think that China also wants to control rare earth refining too. Soon, China could also dominate the market for processed rare earths.
The reason why China supplies 97% of rare earth metals worldwide is because of cheap labor costs. Mining rare is much cheaper than, for example, in the US.
We have become overly dependent on Chinese exports. Entire industries cannot function without rare earths anymore. Without them, the lights will go out, quite literally. Because, for instance, energy-saving light bulbs contain rare earths. The governments of the US, Europe, and Japan have filed complaints with the World Trade Organization against China and are making plans to look for new deposits around the world.
Geostrategically, it makes sense to protect your own mines and not to ransack your own deposits. For example, the United States have their own oil wells, which they haven't touched yet. Instead, they bought oil on the world market. I would do the same. I wouldn't use my own supplies if I could get the same product cheaper on the world market. And it also makes sense to protect raw material deposits.
Bergisch Gladbach, Germany. China's restrictive export policy affects particularly those who need large amounts of rare earths for their daily business. Like the manufacturing company Maxbay. The company founder once invented the magnetic fridge door. Today, the company mainly works for the automotive industry. A special magnet for each application.
We currently produce more than 50 different types of magnets for our customers. We produce 100 million magnets per year, which equals about 1,000 tons of material, including 50 tons of rare earths.
Nowadays, all modern high-tech magnets use neodymium and dysprosium. These rare earth metals ensure that the magnets are stronger and have a longer life.
The neodymium iron boron magnets are used for sensors in windscreen wipers, interval switches, and in steering systems where they control rotational or angular movements. This makes driving more comfortable.
But expensive or failing supplies of rare earths make life difficult for the magnet makers.
We noticed that the shortage triggered speculation. Prices for neodymium oxide rose rapidly. They exploded. They tripled, even quadrupled within 2 or 3 months. We had to shut down production and couldn't deliver on time.
Missed deadlines and exploding costs. The crisis hit the company hard.
Some projects had to be canceled. The industry desperately tries to avoid this dilemma of expensive raw materials and failing supplies.
What has happened to the magnet makers is happening to many other industries too.
The entire rare earth consuming industrial sector in Central Europe and North America is suffering from the twin problems of high prices for raw materials and a strategic dependence on a single supplier, in this case, China.
Village, Germany. Right here, in idyllic Saxony, an inventor tries to help the high-tech industry. Chemist Wolfram Palich is working on a method to recycle rare earths from industrial waste. He enters uncharted territory.
Here on this plate, we have waste from the production of permanent magnets. Iron plays an important role, but also rare earths such as neodymium, dysprosium, and praseodymium. In principle, we're now trying to isolate these individual substances.
The magnetic waste is first heated in a blast furnace, the first step in the recycling process.
There are numerous types of high-tech and green-tech waste. We know that during the production of magnets, a lot of waste is produced, and we've been started to develop recycling processes for this waste. we separate the basic components to get pure, uncontaminated rare earth compounds, which can then be recycled back into the economy.
The recycling processes that exist today are usually only capable of extracting waste material. But Wolfram is not happy with that. He wants to recycle all the materials contained in the magnet.
It's not enough to just recycle gold from electronic waste, for example, if you then discard all the other ones.
It makes perfect sense. Instead of mining and processing rare earths with environmentally damaging techniques, we could simply recycle them from our waste. But the technology is still in its infancy and doesn't work for all rare earths. Alex is currently working on the process of recycling the element neodymium.
We basically extract all the water. This is done in a rotary evaporator. In the case of neodymium, dry substance undergoes a color change and goes from green to blue.
Here in the lab, the procedure is already working. But can Panic make his recycling process work on an industrial scale? Could his method solve our commodity crisis? It's definitely the right idea. We all know that petrol cars are outdated. Electric vehicles are the future. But their engines need strong magnets with lots of rare earths in them.
When these devices are operating, they actually generate a fair amount of heat within them. Any inefficiency in the motor or the wind generator is realized as heat within the machine. And so they have to operate at temperatures much higher than one might expect. And without dysprosium, this is not.
Dysprosium makes the magnets of electric car motors heat resistant, but it's expensive and environmentally damaging. Therefore, car makers would rather not use it. Ames, the United States. Here in the small university town of Ames in Iowa, Bill McCullough set out to revolutionize the design of electric motors. His research is revolutionary, but the US government decided to get involved in the project. He and his team analyzed one of their latest magnetic compounds with an electron microscope. For the electric motor operation, the town needs a new magnetic compound that doesn't contain expensive raw materials. They would prefer to replace dysprosium with silver because it exists in abundance. Unfortunately, the magnetic properties of cerium are quite. However, the secret is in the mixture. To start with, the compound is heated vigorously.
Dysprosium is a huge problem. It's a problem for the Chinese, and it's going to be a problem for the rest of us because we use dysprosium at a higher rate with respect to neodymium, praseodymium, the other rarities, than its natural abundance.
The freshly mixed magnetic compound now has to be prepared for analysis. It's reheated and poured onto a rapidly rotating wheel.
Light be with you.
A vacuum prevents air from
For the Alliance.
the compound and contaminating it. The result is an ultra-thin metallic band that breaks up into small pieces. These pieces contain only tiny amounts of the expensive element, dysprosium. Scientists still hope it makes for a strong magnet.
We really only take advantage of dysprosium at certain locations around the magnet. So if I have dysprosium at the very middle of the magnet, it's not going to be any good. Is being developed dysprosium only where it is required. This can greatly reduce the amount of dysprosium.
In the laboratory next door, shavings of the new mixture are prepared for analysis. The metal strips are fixed on a specimen holder with glue. For Bill McCallum, every new compound is an attempt to get closer to the perfect magnet. Super magnet is a difficult process that takes a lot of time and money. However, the involvement of the US government shows that politicians, too, are starting to see the importance of such projects for our future.
Now we're seeing that there are various trade-offs that we can make, and we believe we still have the performance necessary for use.
The production facilities of the American sports car manufacturer Tesla. The small company only produces electric cars and doesn't use any rare earths in their motors. How is that possible? The secret lies in the design of the motors. They work without conventional magnets.
Well met.
Other manufacturers
Bless you.
similar devices.
One of the things we're observing, or have observed over the last 2 years, this is spurred by unstable has been an effort of the building users to re-engineer their products away from the price for the magnets to a certain point. It's very important, and we're going to
The international car manufacturer started cooperation with a small California company. The idea, getting rid of rare earths in all electric vehicles, even in expensive everyday cars. Zero emission power plants. Wind turbines are spreading quickly. In 2020, they will account for 6% of electricity generation. This equals an output of 1 new power plants. latest large-scale offshore wind turbines are very powerful and require little maintenance. But they work with large magnets that contain huge amounts of rare earths. A massive problem for the environment.
In general, rare earths provide alternative energy generation. You could say that renewable energy generation requires non-renewable raw materials, and rare earths are a prime example. A 5-megawatt wind turbine, for example, contains 800 kilograms of neodymium and 200 kilograms of dysprosium. In total, 1 ton of rare earths for a single 5-megawatt wind turbine.
The largest emerging market for wind power is China. Here too, manufacturers rely on rare earth metals for their turbines. The latest models need up to 4 tons of the valuable raw materials, a huge strain on resources. Würzburg in Germany. Researchers at the Fraunhofer Institute for Silicate Research are interested in the massive amounts of rare earths that are used in wind turbines and electric vehicles, but also
King's honor, friend.
tiny amounts found in computers and mobile phones.
What can I do for you?
They see today raw material rare earths.
Go with honor, friend.
How effectively do we recycle rare earths from our waste? We don't recycle them at all. This has something to do with the fact that in most devices, rare earth metals are used only in tiny amounts and in many different components. Our current recycling methods can only recover standard materials such as copper, not rare earth metals. So we need to separate the different components and then concentrate and get out the rare earth metals.
Recycling devices such as mobile phones because no one knows the basic material, not even the manufacturers themselves.
Hey there.
Suppliers in turn get their raw materials from other suppliers.
First, we need to know.
See you later.
Right now, we have the data for various materials.
What can I do for you? Have a good one.
Once we know it, then we can start selling currently, simply not enough to extract small out of a big pile of electronic waste, like mobile phones.
With a scanning electron microscope, researchers hunt for even the tiniest amounts of rare earths. If we want to we need to get down to the nitty-gritty. At molecular level, the discarded gadget reveals its hidden values.
If manufacturers realize that they are wasting valuable raw materials, if they stop thinking about the money they get simply selling products, then they will understand this old device is a source for making new devices. And then we start to have a recycling economy.
The electron microscope shows the basic appearance of the device. The grey patches the computer only takes a few seconds to find out what the small grey spots are. They contain neodymium, dysprosium, valuable rare earth metals. But it's still a long way to the recycling process.
Who has the chance to lead the way in recycling? We need these raw materials for our economy. Our industry needs it. But our countries are poor in natural resources. the other hand, we've developed recycling methods in the past, so we aren't starting to experiment. And I think this is our chance. I'm not only talking about recycling rare earth metals, but about recycling all materials, including copper and aluminium. I myself am working on recycling raw materials from old waste disposal sites. And it's a huge opportunity for Germany and Europe. We, as citizens and as consumers, need to be aware of the products we use. What is their commodity value? We often discover more than 10 or 20 mobile phones in a row. Usually they're 70% rated. This is no longer sustainable. A cell phone is like a mine. We speak of urban mining.
Good day to
We
you.
need to recycle it. This is important.
Safe travels. How are you? See you around.
Recycling is only economically viable if prices for recycled raw materials are lower than the prices for primary raw materials. At the moment, this isn't the case. So we're now developing technologies that will be used in 5, 10, or 15 years. If commodity prices continue to rise, it'll be profitable one day.
The Americans have reopened Mountain Pass, which used to be the world's largest rare earth mine. Rising prices on the world market drive the project, and the prospect of independence from Chinese suppliers.
Need help?
The investors hope for a 15 to 20% global market share at stable prices.
Be careful.
They use sophisticated technology to protect the environment. The mining company promises to use cleaner processes in the future than in the past.
Hey there.
And still hopes to be cheaper than the all-powerful Chinese competition.
Safe travels.
And the processes that are typically used in China,
How are you?
they use acids that are so aggressive that they chew away on the chlorine oxide in the presence, and it literally goes to sleep. So, for instance, if you take sugar and you put it in water and it disappears into water, that's dissolving, it's going into solution. So these strong chemicals will do the same thing with thorium, and the thorium ions go in solution. Then it becomes wastewater. And our system, we have redesigned our chemistry as such that we avoid the solution. So it stays inside. It stays as solid, and we just pull the rare earths away from it. It took a while.
Rare earths. However important they may be for the future of our modern technologies, their environmental problems are hard to control. Yet many experts believe that these difficulties can be overcome.
So our goal is to be the true rare earth innovator. The most innovative ever. So we're going to continue to new platforms.
But still, aren't all those tons of rare earths used in electric cars and wind turbines the environmental problems are supposed to solve?
At least for the moment, outweigh detriments. Obviously, we have to do everything we can to keep it that way. So we must work hard to ensure that the use of these elements doesn't have any adverse effects.
We all know that the supply of raw materials is dwindling. We
people thought you were a fool when you talked about commodity prices. Today, everybody talks about it. And everybody talks about rare earths. We need a more sustainable approach for producing our food. We need one for sourcing our raw materials.
Sleeping on? The Serta Arctic mattress keeps you cool and comfortable all night long.
What is it like? What makes it different from the everything, living or not? These fish and the water the fish are in are just soup. What makes the fish alive, not the water, is how the atoms are organized. They're special. The double hit, or the thousand chemical alphabet A, T, E, and G, which in different combinations make flour, or a cell, or food. Every living creature has its code. Grow, produce. DNA turns out how it does that. Here's the mystery. 20 years ago, scientists learned to read beginning to end as genes. They did that for simple things like bacteria, and for more complex things, and eventually for humans. And then they began to take the globe and transfer it to make critters do what they'd never done before. And as biologists got better at this, a new kind of science was born. What can we do? Well, we can improve on what's been spelled out over a billion years of evolution. Take it beyond reading, get it. Our own ideas, making features drastically different and even if How do you do that? Well, already, one group of scientists have rewritten and rebuilt the entire instruction with a very humble little piece. They plan to put their new recipe. Links every feature back to the first discontinuous. Take this new introduce my panel. Really studied panel, I gotta say. Particularly for a Saturday afternoon. It's like this is a Saturday night, 9:30 kind of group of people. So you're lucky here. So let me start. First of all, and tallest, at 6'5", is a professor of genetics at Harvard Medical School, a highly prodigious engineer. Many times over a businessman. He's founded I don't know how many.
Good day to you.
With wonderful names, Veritas Genetics. Like I said,
see you around.
Gnome, not spelled with a silent G, like gnome, but weirdly with a K. Gnome. Ab Vitro, N involved, Gen
Well
9,
met.
and a solar fuel company that I really like, Unlimited. He is a leader in the field of synthetic biology, won all kinds of awards for excellence, elected to the National Academy
For the Alliance.
and to the National Academy of Engineering, and he gets an enormous amount of attention, often admiring. When he went on the Stephen Colbert show, Steve greeted him by asking, by a killer virus or more like a giant mutant killer squid man who arises from the Pacific Easter Island entity and eats all our planet? They will be asking such questions. Dr. George Church. Also with us is a professor of biochemistry and systems biology at Harvard Medical School, the first director of the Harvard University Graduate Program in Systems Biology, and as of this week, the proud co-parent, I'm Andy Sarah, of what is called a biology. This is a device that, like a tree, uses sunshine to split water into H's. H's actually are then fed to a starved bacteria. The hungry bug will gobble the hydrogen and turn it into, of all things, fuel. So these kind of
Light be with you.
Sunshine and water, these fuels will one day be very, very cheap. This is
Be careful.
The co-founder of a company called Dickus, which, as its website sells engineered organisms that make the world better. Not only I started life as a computer geek. In fact, he started very early. He spent his high school summers at the Artificial Intelligence Lab at MIT back in the 60s. He was involved in developing ArtNet, and then sometime later on, he was reading Harold Marowitz's He got interested in the idea that I was now often called the godfather. Switch. And standard blocks of DNA that contain basic life. So, like Lego, like Lincoln logs, you might build all kinds of new kinds of features in biobricks. And every year there's a competition, and from all over the world compete using bricks to build God. So, bringing doom to our species or salvation and good health to the rest of us. Either way, let's welcome Tom Knight. And also an assistant professor of bioengineering at Stanford University. He is president of the BioBricks Foundation, which they're linking to Tom. Drew also runs the BioBricks iGEM competition for students all over the planet, and he's also the author of Synthetic Aesthetics, Investigating Synthetic Biology's Design of Nature. That is Drew Endy.
Hello.
Okay, so just to make sure that everybody's on the same page, I'm just going to do something a little bit basic just to begin. This, of course, as you recognize, is a little...
Have a good one.
It's actually signed, I don't remember exactly where, but in my fanboy period, I got Jim Watson to sign it, and then in my PC Now Dementia period, I can't remember where he signed it. But anyway, this is a soupçon of a DNA molecule, and what we're talking about the A's and the C's and T's and G's, of course, we're talking about these sort of inner steps. So my first question is, I'm wondering, where do you find these things? If you want to build life and you need to have these chemicals, where do you go? Is there a store? Or do you have to go to a living animal and suck it out of it? Hey, the internet. No, there is nothing. The internet is something you look at.
If I could get your credit card and go to the internet, I would go to a website and start typing in letters from A to Z. taken from the milt of spawning salmon. And it would be deconstructed and then reconstructed. But when it became interesting to the pharmaceutical industry to make some medicines that were based on the basis of DNA, they were a little bit nervous about the reliability of the salmon harvest year after year. And so they invested in developing new synthetic chemistry that would start with sugar, often from sugar cane, and derivatize that to make the raw individual bases of DNA from scratch. So not the, if you were to get this DNA, you're not starting with strawberry DNA or lizard DNA. You're starting with bases that are coming from sugar produced in sugar cane.
Okay, so if I want to make something, Pam, one of these things wouldn't get me very far. Like if I wanted to make a gene which would spit out a protein, which is what we're made of, so I could make something real. How many of these would, I don't know, if I were trying to make insulin, how many of these would I need?
Wouldn't need that many, a couple 100,000 or so.
A couple of 100,000?
No, a couple hundred,
or
up to 1,000.
Okay,
and
yeah.
that's totally manageable. We do that all the time. I can design a protein, as Drew said, order up the DNA, get it delivered to me. This is standard procedure now.
So you'd go to, so if we wanted to make insulin, you'd look up insulin, and then it would say, okay, recipe for insulin, A, T, D, 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 and there'd be like 100,000 letters or 10,000 letters or whatever, and then you'd send them to DNA or us.
Well, we have an enormous resource out there, which is called the internet, as Drew said, where we can go, the information's all there. I want to make insulin, I want to make growth hormone, I want to make lots of proteins that could be useful. That information's out there, and anyone can take it and reprint it, order the DNA. The skill set becomes putting that DNA back into the organism and getting it to perform.
Okay, so just before we take that step, let me just run a quick video that you can watch. This is what happens at the DNA or Us store. Like you can get, that's T, one of the letters, and it's in a little bottle. And I think that's me a few years ago. I'm going to pour it in my hand. So you can see it's a powder. My finger is going to so this is not, doesn't look very alive. It just, and here's the other letters. So now we're going to introduce you to a robot, which is color coordinated, and we'll just basically spit out the chemicals in the exact order, zip, 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 like that. I don't know what this is. Probably some other part of it. And then that's the deal, right? So this is a robot. This is like a factory. Yeah. Yeah. All right. So what I guess I'm wondering is, once you've got it, let's make sure that is not yet alive, right, George? It's just at this point just organic chemicals. Right. Okay. So now, once we want to buy some, just so I get a sense of the cost. Do you have any idea how much it costs to make, I don't know, like one pair?
It's on her friend.
By the absolute lowest cost method, which is not the one that we chose, but is on chips, you can get 2 million of those pairs for $1. That's raw, fairly short pieces of DNA.
Okay, so this is so just to make an insulin gene, it's going to cost us about $50. Let's take a look at the next thing we're going to do. Oh, we're going to go and do a whole chromosome. That's 135 million of these things. So that's a $4 million. So that's not George's price. That just seems to be, we seem to be a little bit, and let's build a human. So it's we're 90 million.
That estimate for the human is both too high and too low. So it's a couple $1,000s just to build it in short pieces. Right. And we don't actually know how much it would cost to produce a fully functional one.
Okay. But so these are like not crazy numbers. I mean, the moon shot, going to the moon cost a lot more than this.
Oh yeah.
See you around.
All right. So now, when what does the
I supply only the finest
string
goods.
it together, or they just send you 40,000 bottles?
So the process starts with making fragments that usually are about 100 or 200 bases long, and then those are stitched together into larger fragments, usually at this point somewhere in the 4,000 to 10,000 base region. then those fragments in turn, those are usually what you would receive from one of the manufacturing companies.
And you get it you get it delivered by FedEx or?
Yes, exactly.
It comes in a little.
Yeah.
Is it like a thimble-sized package, or is it like a box?
Usually we get a box, a styrofoam container, very similar to this one. In fact, this may be one.
There's a giraffe in there.
Yes, which has usually many different versions of these genes in them, hundreds.
Typically. And are they in liquid form? Like are they is it a paste like toothpaste?
Usually it's a solid in the bottom of a tube, which then we redissolve.
Okay. So you'd get like a string of a lot of them in the right order.
Yes, that's.
Does it come with a letter called "We've been very careful here, and our robot acknowledges that this is exactly."
And usually they are sequenced after fabrication, so that you are reasonably certain that they are correct.
So then you have to put these segments in the higher order in order to get.
Yes.
Okay, so now let's. But we're still dealing here with something that is lifeless at this point.
Absolutely.
Just dust. Okay, so let's turn the dust into life. So Craig, this other guy, he found this very simple organism. I think in the ear of a goat or something. But anyway, it was a very small living thing, practically a virus. And he copied its A's and C's and T's and G's, and I guess he stitched it together in this form. So he ended up with the full spelling of this little thing. Now, we're going to do something. We're going to plop it into something. I could say we could plop it into a waiting room. We could plop it into a suitcase and slap it closed. That doesn't seem... We could plop it maybe into a computer, which has a hard drive, and so you pull out the software and you plop this new software in. What would be the analogy to describe what's about to happen that you'd like?
Well, if you want it to act like life, it has to go into a living cell. You can, in the case of the situation you mentioned, you can replace what's driving the life of that cell with the artificial synthesized piece of DNA. And it can take over and allow that cell to grow.
So you scoop out whatever's in there, you put in whatever you want.
Right.
Will this, will it boot up?
If you're lucky. I mean, I think this is the challenge that we're talking about here is how do you go from this key step of being able to put things together then to getting it to boot up in the cell. And I think the experiment you talk of took something like 15 years to boot it up, and it didn't work the first time. And I think...
It didn't work the first many times.
Many times. And the nice thing about life, though, and I think Drew also said this, is that it either works or it doesn't. If the cell doesn't grow, you have a faulty system. And there's only one definition of working here.
But the empty cell that is still still fertile, I guess is maybe the word, wants to grow something. So is it indifferent? I mean, if it has just been a grape cell, you can't make a grape cell. Well, you could. Well, let's make it a zebra cell. If it's just been a zebra cell, you get rid of its zebra-ness and you stick in whatever you've just designed. Won't it say, I am not that, I am a zebra, I reject you, I won't, or does it not care what's in it? It's an omna ovum.
It depends on how far away it is. So if you, most of the sorts of experiments you're talking about is either scooping something up or pushing an extra one in and destroying the other one once the new one comes in, they have to be very closely related. The further away it is, it's basically, the contents of the cell are programmed by the previous DNA, and if that's incompatible with the incoming one, it will just get sick and die.
How do you, if the thing succeeds, like if you have a human baby and the baby is alive, it will cry. You'll know, oh, it's alive. What do these things do that makes you think, oh my God, this worked? Like, do they have a baby? Or what
they reproduce.
They make more
copies. Reproduce.
Yeah.
They make more of themselves.
They make more of themselves. So that's got to be a pretty cool moment. Like, you sit there and thinking, okay, and then you wait, I guess if it's a bacterium, you wait a full 20 minutes for it to get old, die, and have a baby, or split in half. How long does a normal bacterium live for, just from hello to goodbye?
Well, they often don't say goodbye. They just keep replicating.
That's right.
Until you kill them. But
so as soon as they split, then you know you've got it. Then that's
exactly.
your moment.
And the fastest that goes is 11 minutes.
So this man, Craig, he got the thing to separate, and so now the next, a few calendar years pass by, and suddenly, this community is interested in yeast. If you can do something and make a bacteria out of dust, why would you then? What's the point of moving on to yeast? In what ways is yeast different? You've hung out with yeast.
Yeast is great for making beer, whereas bacteria is not.
Or bread.
Or bread.
We're all very familiar with yeast. In fact, it's the world's oldest industrial organism, probably. The difference is that bacteria are what is called prokaryote. I think they teach this in elementary school now. They have, in general, one piece of DNA called their chromosome. Yeast, on the other hand, are what's called a eukaryote, more like cells in our body, where their genome is separated from the rest of the cell by something called a nucleus. This is what distinguishes higher forms of life. Moreover, they have more than one version of that chromosome. They have multiple different ones. And this is a key property of higher cells.
So if you would a eukaryote, this more complex cell, it would have like 10 million base pairs or 12 million or something like that, as compared to the few hundred we were talking about before. So it's more.
Yeast is about a 12 million. Bases.
2 chromosomes for a bacteria. How many for a yeast? Something like.
Depends on the yeast.
Okay.
It's an interesting question, by the way. Why do some yeasts have only a few and others have a lot?
You've asked them, and they're not telling you. Why is this becomes a world? This project is apparently so complicated that you need scientists in the United States, this guy Jeff in NYU, and elsewhere in America, and China, and France, and Australia, and Germany, and Singapore, and the UK. I'm just curious. Is this is a big deal to get a yeast from the beginning to the end, boot it up, and make it reproduce? Has this happened yet?
No. But just to be clear, they're not doing the booting thing. They're not scooping out the yeast nucleus and putting in a new one. They're editing big chunks at a time. So they're kind of continuously evolving the yeast by design to a new one. The end product is a completely different. Well, not even that is completely different. They've changed certain things along the way, but they've synthesized it all.
It's a newer, slicker, designed yeast once it gets.
Isn't it called yeast 2.0?
Yes.
Yeah.
It is.
Yes.
Okay.
Yes. One of the nice things about yeast, very much unlike bacteria, is that they have sex, and so one can breed, these yeasts and mate them.
Suddenly, I'm so much more interested in this.
Right.
Beer and sex.
Oh, beer and sex. Yeah, like you could be a frat boy in this kind of a business. Okay. So they have sex. So that means that it's not just a splitting in half. This would be a reproduction that you could applaud.
Yes.
And the reproducing could even be fun for the yeast. Okay, so
one of the things about the project is not only scientifically and technically complicated, but it's sort of reflective of the importance of doing things together as a culture in synthetic biology, that people working together around the globe, learning how to do the new things, so that biology's everywhere, right? So as we get better at the doing of new things with biology, bringing people along together is super important.
There's actually an interesting precursor to that in that the sequencing of the yeast genome 20 some odd years ago was also a distributed effort where different countries took different chromosomes. So there's precedent for this kind of model there as
but
well.
if George says if there's some tinkering going on, like do the Chinese and the Romans and the folks in Nice all agree, or do they quarrel over this yeast that they're designing?
No, it's a true consortium. They've set certain goals. So, for example, one of the goals is to collect all of one kind of gene in one place, or to change one codon of the 64 codons throughout the whole genome. So if they each picked a different codon, they wouldn't accomplish their goal. So they picked the same one, which is UAG.
UAG, okay. So when the yeast project, and by the way, just to go back to that bacteria that we discussed before, when Craig finished with the bacteria, it was a copy of an extant bacteria, but he apparently added some language to it. Do you remember?
He added his name.
He added his name.
It's like graffiti, right? It was a type of graffiti.
It's graffiti.
He added. So how you mean you can take the alphabet, these alphabetical pairings here, and spell out words like J. Craig Venter? But this is A, C, T, and G, so this must be some translating device or something.
A decoder ring.
George, you've had a certain personal history with this. For some reason, you are fascinated by writing in genetic alphabets. You wrote a book in English, because you are an American, and you wrote it in your native language, and then you were seized by an urge. What happened?
Well, not just the English text, but the photographs as well are all zeros and ones in the computer. And so that's not too far away from the C's, A's, G's, and P's. So we encoded it from the zeros and ones to the DNA and made 90 billion copies of the book.
You put your whole book into what exactly?
Into a 70 billion copies of the book was so small that it's just a speck of dust.
Does that make you the most published individual in the history of...
Yeah.
If at least unread, but most published, I guess.
We did read a sample. We read a sample of the books, the whole book. We read the whole book, but just a few of them, a few of the copies. So some of the copies are almost certainly wrong. Yeah.
But this is like a strange enthusiasm. I remember, maybe I forgot to ask you this before, but is it true that you once actually had to review something for some fancy medical journal and decided to send the review in A, C's, T's, and G's?
Actually, it was Craig's genome that he had put his name in. The code that he used for his name, I didn't think was the ideal code. And so I made up my own code, and which I thought was more general. So his, for example, was all uppercase, which on the internet is yelling. So I had one that had lower.
Craig Venter! And
Yeah.
you want them to go, Craig Venter?
Yeah, ideally, yes. And punctuation, and also photographs were missing, so I wanted to have photographs. So that's just a simple 0 to 1 to ACDP. I sent this to the editor, figuring that she would tell me to translate it into English. She sent it right on to them without reading it. And then they had to figure out the code.
So they tried to figure out what you'd done.
They did figure it out.
Okay. Yeah. So this is translated.
Clyde Hutchinson figured it out how to do it. Yeah.
That was smart. So let's now imagine that we have, or about to have, or one day soon we'll have a synthetically built yeast. I'm just trying to figure out what we might want to do with it. For one thing, somebody here, I guess you're at Ginkgo, right?
Yes.
Yeah. So you got an order for a, this is an actual rose. Yes, it is, an actual rose. You got an order from a perfume company or something?
Yes, there's a company, a French company called Robertet. They've been in business for about 200 years. They extract rose oil from natural roses.
Is rose oil something, when I'm smelling the leaf, that's what I'm smelling?
Yes.
Is rose oil an abundant thing that you can have by the gallon?
No, not typically. They grow the roses in Turkey typically and import them to France. The rose oil is extracted and they use it in perfumes.
And so this rose would have how much rose oil?
I don't know,
but it's
not very
itsy,
much.
bitsy. Right. So if I'm a perfume company and I want to make a rosé or don't know what perfume rose products are. Eau de rose.
Right.
If I want to make eau de rose, so I need a bunch of rose oil.
You need a lot of it.
So your thought was maybe you could build a rose cow, like if you were a dairy farmer, you know how to get milk. So you thought you'd.
It's one way of thinking about it. The way I think about it is that the way I think about this is that fundamentally biology is a manufacturing technology. Biology is all about making more of something, converting one substance into another. In many cases, it's converting food into more copies of itself, or and we use that ability all the time. We ferment, barley into beer to make alcohol.
Right.
In this case, what we're doing is, using that same technology, very similar sort of technology, to convert sugar into the same chemicals that would be, present in a rose.
Well, do you go to a rose to find out where rose oil comes from, or do you go to anything you can think of?
Well,
like is everything you need to know here?
Pretty much, it is. Although, so we do an analysis of the genome of the rose, typically not looking at the DNA, but rather at the RNA of the rose, which tells us much more about what specific proteins are being made in the rose at specific times. And then we try to recalculate, what the chemical pathway for making those compounds looks like on the basis of what we discover in the rose, the information which is present in the rose.
But how long a conversation if you ask a rose, say, how do you make the rose oil? Wait for the answer. Are you waiting for years? Are you waiting for months?
That's a challenging. I mean, that's a scientific question, and one that people have worked at, for a number of years. So it's one that's already been studied. And
so you just go to the internet again.
You can go to the internet. Yeah.
And does the internet say, okay, so to make the rose, but you want to hitch the rose to your new cow, which is going to be a yeast.
Yes, it is.
Do you have to, you're going to have to get the yeast comfortable with making something it's never made before.
That's right.
Is that hard?
Yes, it's challenging. And, there's a lot of challenges.
What are you looking
there.
for?
They,
I supply only the finest goods.
Doing what they always
I buy
do,
and trade.
Making more yeast.
Need help?
And,
well met.
So
forget something.
Very often, in
I'm
addition to adding some of pieces of DNA.
What's your problem? You're getting on my nerves.
That you care about.
Knock it off.
You're also,
King's
in
honor,
many
friend.
cases, eliminating, some of the other mechanisms
for something
that
specific?
the yeast is using to do other things, like make alcohol.
So you say to the
Farewell.
yeast, stop that, do more of this?
Exactly.
And then when it's time, have you done this?
Oh, yes.
Did you get the yeast? Did you, I shouldn't use these words. Did you implore the yeast, or enslave the yeast, choose your verb, to do this so that it begins just spitting out the rose juice?
Yes.
So I happen to have, because you gave them to me, Rose B and Rose A. Now, does this mean that the fact that I have two tubes of God knows what mean that there are many roots to the rose, or does this half of it and the other half is here?
So rose actually is not a single chemical. The scent of a rose is actually probably around 200 different chemicals, and everyone probably is aware that different roses smell different.
Right.
And so, depending upon what kind of rose you are trying to synthesize a scent for, you have to make a different mixture of those chemicals. So in this case, what we're doing is making individual chemicals in each of these, and they will be mixed together to form the correct scent. We will eventually be making versions of rose that make many of these chemicals at the same time.
So if Pam, you want to do this? Because
I actually
frankly, a little bit
that I can try. Well, this will be a challenge to see how strong
this
it is.
Yeah, see if you can smell it.
Oh, I can. It smells like yeast.
Yeah, probably.
Okay, well, that's going to be a problem later on.
This one's a little better.
Yeah. Yeast does smell like yeast. It likes to do what it does.
Well, I'm reminded actually of
give it to
a
George.
famous experiment done by the MIT iGEM team, OD coli, where E. coli actually smell terrible, as most of us know. And Drew, I think, Tom also led the MIT iGEM
Do you smell rose?
I like the bee better. Yeah.
But didn't they smell like mint? You made minty
fresh and banana.
And it was what was it called? OD coli.
OD coli.
Yeah. So apparently, underlying all of this is this sense that you will be able to make up, maybe even outrose the rose once you get this thing going.
Oh yes.
Oh,
I think
yes.
so. I mean, one of the things I think I mentioned to you earlier is that the natural rose oil has a human allergen which has to be removed before it can be sold, and we will simply avoid making that allergen.
I'd like to add one other thing about this technology, because if you're making rose oil, and Tom, you can correct me if I'm wrong, but you generally have to use a lot of sort of nasty chemicals to extract it from the rose petal. And much of what we around us uses petroleum-based chemicals, whereas if we use life like yeast to make these things, I think we have a cleaner chemistry. Also, as Tom is alluding to, the cells themselves are great chemists. We can learn from life as chemists, they're probably better than the chemists in the laboratory. So I think these are some of the things to think about going forward in terms of a living chemistry.
Well, let me go to another chapter of tinkering. This is a George chapter. It's just kind of interesting. If this E. coli is a sort of workhorse in the laboratory, one of the questions that scared and timorous people would have is that if George or any of you guys engineers an E. coli that is new to the world, and it just slips out the window and goes, I don't know, out of NYU into Washington Square Park,
If you find a
that
powerful
it might
weapon,
need another.
just make sure it isn't corrupted by the burning legion or old gods.
But because bacteria, it would just kind of slip something from it into its wild neighbor, and the neighbor would either die immediately or create, perhaps in some occasions, an advantage which would maybe affect E. coli everywhere. So I don't know whether you thought, okay, this is somewhere coming at us, or whether you were just worried about safety, but you began to think about this, and what did you do?
Yeah, well, we worry quite a bit about safety. More than worry, we do in safety engineering, which there's almost every field of engineering has a safety component. And in this particular case, most organisms you deal with in the lab or even on the farm, if you put it out into the wild, it will disappear. It will be eaten and die and not reproduce. But we were making a particular one where there's an industrial problem with viruses contaminating your process. Even one viral particle can wipe out your factory for 2 years. So we wanted to make a virus-resistant E. coli to solve that problem. But
so that's the opposite. So if the window is open in the lab, in comes a virus, it lands on one of your E. coli, and your entire lab goes down.
But that creates a possible new problem, which is that now it could live in the wild. It could outcompete all the other E. coli, hundreds of viruses to which they're sensitive. This would have 0 viruses to which it's sensitive.
I see. So the mutant E. coli, having just gotten virus resistant inside the lab, then lands on the hoodie of Tommy, who walks back into Washington Square Park, and off it goes and conquers all of Washington Square Park and beyond.
Yeah, because E. coli is a global species. It goes around the world quite quickly. We don't need to get into details of how it goes out. Yeah, and so we wanted to make it so as soon as it left the lab, it would immediately start starving for something that's only found in the lab. In the past, people have made it so it would starve of something that might be in the wild, though we felt that wasn't safe enough. We wanted to make something that was only in the lab, so we made it addicted to or dependent upon a particular, not laboratory-derived chemical called BIP-A, It's a large amino acid. It's larger than most, or larger than all the rest.
But this is like a gruel or a broth that it sits in when it's
it's part of the broth you grow it in, right? And it's inexpensive, and it doesn't need very much of it, but it does need more than zero. So the standards, such as they were that you needed to have something that would not escape at sort of 1 in a million. But 1 in a million is basically escaping. So this one we've clocked at about 1 in 10 to the 16th. So it's about 10 orders of magnitude better. And it could be even better than that.
10 to
Just gets hard
16
to measure.
zeros? I don't even know what the word for that is.
Yeah.
A gajillion.
Brazilian. Yeah, so
wait a second. So you've addicted this, you have this factory animal for your yeast. You tell the yeast, well, which is normally eating lamb chops, potatoes, and salad. No more salad, no more lamb chops, no more potatoes for you. You're eating this rarefied gruel. That's all you ever want. And if you can't get it, you die.
Well, they get all the other stuff too, but they need this new thing, or else what can I do? When they get on the wild, they won't find it. Or even in the laboratory.
If they get on the finger of me, I don't have that gruel on my finger, so they would die on my. How long did you decide to let them starve for?
Well, they die almost instantly. We have done one experiment where we slowly reduce. This is the people for the ethical treatment of bacteria probably is, but anyway, slowly reduced it. So now they actually need much less of it, which makes it less expensive for us. But we could never evolve them over many months. We could not evolve them to the point where they were truly independent of this amino acid, which is a good sign that it could be stable. But to really do this with
So Drew, I'm just wondering, this got a lot of attention, this thing you did, because apparently it was a very, very elegant piece of engineering. But is it very hard to take a yeast and change its digestive system? I don't know, why was this such a big deal?
Well, it's a lot of work, but it's a big deal because it addresses one of the concerns that arise when people think about deploying engineered biological systems into environments that might be outside of a controlled, enclosed environment. I think to jump back to something, though, the word creation has a lot of implications. So as an engineer, I'm not familiar with the implications of the word creation. It implies I have an unlimited budget. It implies I have a perfect understanding of the material I'm working with. It implies I have absolute power. I can do whatever I want with the atoms. I have a godlike power. As an engineer, unfortunately, that's not what I experience. I have a finite budget. I have an imperfect understanding of what I'm working with. I have a limited capacity to deploy my intentions into the material. And yet we still struggle to realize things that behave as expected. So, I grew up and was a civil engineer and worked for Amtrak for a time. And so I'm familiar with, hey, we know enough about gravity to make a suspension bridge, and the physicists can still be doing the gravity wave detectors, and that's all great, but we got to make the suspension bridge work, right? And so having an operational understanding sufficient to get reliable behavior is a big deal. When you're interviewing that student, what they're reflecting is this culture of just dealing with our limited capacities, yet still deploying something as responsibly and reliably as we can. And so to do the hard work of making a best available technology as a type of genetic firewall or fire break is great.
On the other hand, it may be that you've just made a yeast which is really fundamentally different from, not just casually, not just perfumally different, but deeply different from, E. coli that went before. You've even told the Times it's almost a new kingdom of life that would have trouble exchanging genetic material with any other life form. So you've made a little itty-bitty, very useful, keep us safe Frankenstein, kind of, right?
Sounds like an
I'm assuming you're thinking I'm wrong.
I think that's unfair. I think it's more like you've developed a new language, and so all of a sudden the people in France are speaking French. And one of the things you can do because basically the way the DNA is being read out has been changed, right? So the expression of the instructions has been changed. And so the way I might offer for thinking about it is we got a slightly different language, and that implies you could have a translator, right? So you could bring in, if you needed to, a way of translating the new language in the new context, but it's just a different language, and if people never bother to learn a new language, things will stay apart, never interact.
Well, let me give you a chance. Jack, are we proud of what we did, and why? I mean, obviously you solved a real problem, but as an engineer.
Well, I mean, to some extent, you don't want to be too proud until many, many years later when you've seen whether it's useful and not toxic in practice. You can be proud of the basic research that you've done, that you've discovered along the way how to make a new genetic code, something that's radically different from previous ones. You've made a solid effort, at least, that you can be proud of, to make something that is isolated, because you not only don't want it to survive, you don't want it to take in toxins and then make those toxins super survivable. So I think, that's all worthy of celebrating as a milestone, but it isn't the end product. The end product.
But Drew is pretty serious about the attitude, my wrong attitude.
Yeah. So, but like maybe I want to be invaded by engineered bacteria. So I'd like to drink a probiotic yogurt that is a diagnostic platform, depending on the disease state of my gut. What comes out of me is a different color, telling me which specialist I should go see. That sounds a lot better than a colonoscopy. Maybe I'd like to ingest engineered salmonella that's been made totally safe from an evolutionary perspective. We're breeding safe vanilla. And this will go into my body and be engineered to attack tumors. Like the yeast that make the rose oil are great, but it's still feeding into this centralized, industrialized manufacturing process. I'd rather Ginkgo upload the DNA sequence for the genes that make those odorants onto the web, and then I could download that wherever I am, print the DNA, and put that DNA into the microbes on my skin, so I have programmable distributed manufacturing of the perfume. Why would I ever have duty-free catalogs and things going up in the aisles of the planes to slather on some stuff? It's crazy. So I want to be invaded by engineered microbes.
That was just a plain old advertisement.
How can such a tiny thing prevail against the might of the Burning Legion?
That this is an area that people will find palatable. This idea of not having a colonoscopy or having bacteria that can affect your mood or your ability to sleep or your diet or if you suffer from IFP. I think this is an area that actually it will be one of the areas where I think the interface between engineered organisms and humans, people will be very receptive to it. There obviously will be regulatory issues around it. But I think Drew makes a very good point that
I think Drew makes a good point too.
It.
I just before I put down all my sabers, just to hear George say, like, when I asked Steve, was he proud? He really wasn't. He just said, "This is what I do." When you've done something that any of the things that you just described, I mean, if he were able to give me something like popped in my mouth and out would pour a color-coded Fall Dr. Jones, that would be such a remarkable piece of engineering that I would feel a little nervous. Like, am I going to get anybody's here to feel magical? Are you just all very work-a-day engineers? You're never going to give me any Murray.
That emotion, I think, comes from working with systems that you don't understand. And I think fundamentally, we're at the cusp of being able to actually understand what's going on in these systems.
Good answer.
I'm still on stunned.
You are.
I think you can have a sense of awe even once you have a large amount of understanding. I think we understand how rockets work, but it's still pretty amazing when we send one past Pluto.
I'm amazed when I get on that 80. It's huge. I know how planes work. That thing is amazing.
Well, let me get to something that everybody's kind of curious about lately, which is to build. This is a new chapter in all this. And this is fairly recent. It's described as a bold quest synthesizing a human genome, which is
the Illidari are about to be overrun.
Dash right, that's the right genome. But the phrase that came with that I saw a few weeks ago, is to synthesize a complete human genome in a cell line within a period of 10 years. It was kind of a call to action. Okay, all the sophisticated ones, let's do this. Let's synthesize a complete human genome in a cell line within a period of 10 years. But my first question is, since some of you are signatories at this point, I'm just wondering, first of all,
There is much to be done.
At first blush, like, what? Like, it sounds like, wait a second, are you trying to build a synthetic...
The fate of our world hangs in the balance.
Get this up. Like, you guys. It sounded suddenly like you may be wanting to build a synthetic human being. Right?
You are our last best hope.
I've sacrificed everything. What
So,
have you given?
in a certain sense, my lab has been in the process of doing this, synthesizing human genomes, not human beings, for a decade. And the goal of the project, as it described, was to make cells
Your
just like
artifact
the
looks
ones we've been
powerful.
talking about. Just
Speak
like
with
the
Jason
full
Alari
IREs
if you intend to
used
help.
for manufacturing. Mammalian cells, including human cells, can be used for manufacturing drugs and other therapeutics, antibodies, and vaccines that help save people's lives. Those cells, just like the cells we were talking about before, can get contaminated with viruses. And in fact, this happened to one of the orphan drug manufacturing companies in Boston. Enzyme was protected for 2 years because of a virus getting contamination. So that's an example of one of the reasons you might want to do the same thing we did for E. coli to make them virus resistant. We'd like to make some mammalian genome at the cellular level. That doesn't mean you should ignore all of the other ramifications, but that the goal of the project is to do simple things like that.
But so I just want to make clear that the cartoon distinction, like, we're
If we don't meet again,
person,
die
or just
well.
a cell that encodes the human genome, or something else other.
The proposal on the table was to make cell lines that would tell us that would answer research problems and answer industrial problems like the one I just described. The issue of making
yeast to go into a person. We're like, what, I guess, again, it's a much bigger problem. It's like, why would you go? Why
it's
don't
300 times bigger genome than yeast.
Yeah.
It's not that much bigger.
Well, 300 seems modest to you. I don't know, it seems
well,
like...
considering that the cost of sequencing and synthesis has increased 3 million-fold in the last few years, 300-fold doesn't seem like asking a lot. To bring the price of it down so we can make millions of
I need a target.
Well, just so I can understand, like if you just get a cell that has all of the human, why couldn't you just do it with a mouse, or do it with a worm, or do it with something? I mean, when you go from a yeast to a person, it seems like it's kind of like come on, just. It seems reckless to just jump such a...
We will cleanse the universe in fire!
You're wondering what about me, the deer...
Your Titan gods have abandoned you. Lay down your arms. There is no hope left.
Oh, you want money, attention, talent, and
I'm
that's
going to give that one to George.
Quite frankly, when we did the human genome reading project, I felt that it should be dealing with for the writing. It should be something where we reduce the cost and improve the quality of the technology, and then people can use it for whatever they want. Practically, the mammalian genomes that you choose to do it on might be the ones that are used industrially to produce right now to produce the therapeutics. So we don't need to, the people that are developing the technologies don't need to think that up. They can just query companies like what cell line do you use it? And then you say, okay, that's what we'll start with.
But you decided that maybe...
And human is one of them. Chinese hamster is another one, oddly enough. That's what the Genzyme virus infection was, Chinese hamster ovary cells.
Well, she's got a point. Like if you said, for the next 10 years, we're going to go out and build a Chinese hamster, then probably treasuries and universities has a certain glamour to it, I guess. I assume she's right?
I think, I think for something this important, you don't need glamour, quite frankly.
Okay.
I think you say, we're developing technology so we can do it on any organism. Call it the any organism genome project. It doesn't have to be about humans.
If you want to improve in a human, if you want to do if you want to create more viral resistance, so that people with Ebola and whatever pops out of the forest is squelched in a human. I mean, I'm sure that they have respiratory too, but or you're worried about neurodegenerative diseases in a human. I can't remember where Jim Watson put his signature. Or maybe you want to be able to build human kidneys or human hearts or human livers for transplantation. These are like this is the good stuff. Do you need a human gene to make that an efficient?
We are the Legion! You cannot prevail!
There's a bit of case by case going on there, but certainly the same reason that pharmaceutical companies tend to focus on human proteins to treat human diseases, to doing the stem cell therapeutics, human stem cells. Maybe you might be transplanting organs that combination of humanized animal, but in both cases, you might benefit from having easy access to the whole genome. But I think you're totally on target. Is the reason we're biased towards humans in a pharmaceutical sense is because that's the more direct route to in this case, stem cell and organ cures.
Pam, you're just very politically carefully sitting there with your hands folded. Do you want to join him and say, yes, you're sort of pro a human cell? Or do
I have a different focus in that I was interested in so first of all, I think this is a real learning moment for everyone, because you think about the scale, and all of you can go look at the human genome on the internet and figure out how good is it. Can I actually copy it or not? And I think there's a learning moment here for all of us. But one thing I've wondered is, do I know enough to even build a single piece of DNA that would act like a human chromosome, which means could it divide with a cell in a petri dish? Now, one of my favorite targets has been the synthesis of the Y chromosome, which no one ever talks about. It's actually one of the smallest, and it's not required. Sorry, not necessary. And so it seems to me like an interesting.
To save all mankind.
But there's an interesting question of, do I actually know enough?
Salt the earth!
It goes back to how much am I guessing when I try to make bacteria genomes? And there's been 50 years of research on human chromosomes.
life is dedicated
Can I go through that
destroying
research
the
and see if there's enough known to even make something that would function like a human chromosome? And
so
that's something that interests me.
you're thinking along Harold Morowitz's lines, like that idea that in order to really understand something, go ahead and build it. That's the
Well, I'm not thinking, yes, that is my view, but it sort of fascinates me, the challenge of, and I'm sure we're going to fail.
It isn't enough.
You're sure we're going to fail?
The first time we try to piece it together, it's going to fail.
It isn't enough to just go build it. You need to understand
Azeroth
why
must
you're
not
building it,
fall.
what those features are. You don't, just to be completely clear here, Craig Venter does not understand how that organism works. He built that organism. He does not understand how that organism works, I guarantee it.
One of the super interesting things is the number of genes in the minimal genome that have no known function has increased over the last 15 years.
Yes.
So you build it in order to not understand it for a while, and then I suppose when you shoot yourself.
In the case of the Venter, what Drew said, you learn what you don't understand.
Right. Think about. I don't know if you ever took apart a car. you disassemble the engine on the car, you've got the parts all over the driveway or the yard, and that gives you some sense of understanding of how a car works. But now you try and put it back together, and when you go to turn the key, does it turn over? And how many pieces did you leave out in the yard that you couldn't figure out how to put them back? Right, so that process of learning by disassembly and learning by reassembly, they're complementary. And it's not until you start putting things back together are you forced to confront your ignorance in a very stark way. It's a very powerful scientific approach.
Well, let me just put in front of you, I'm sure all of you would disagree with this, but I think it ought to be said. This is about
I can't attack that. I can't attack that target. I can't attack that.
It only seems normal to consider the possibility of success. So while you can dwell on the possibility of failure and all the learning that will come from failing, let me just throw out what happens if you're going to create a specifically designed toon, then what does that do to the toons that are sort of naturally occurring? Now, there are two kinds of toons. If you can improve something
Your
by a
Titan
design,
gods
why
have
not design
abandoned
your own
you.
kid to be one of those? So the people who
Lay
want
down
to
your arms.
start getting
There is
kids
no
who are
hope
under a year and a
left.
half and always gorgeous and beautiful to look at and get the best girls or the best and are athletes and blah, blah, blah. And the poor people just can't compete. So you get a sort of appreciation by income. Like, or you have deeper still, you have the Declaration of Independence, which is our code of the country. We hold it. All men are created equal. What if you create an engineering project and you created one people? So we're going back to this is just fun. So let's ask, when you guys have you wondered a little bit about the moral consequences or even of the possibility of success in this way?
Well, I don't think you need to go to a synthetic genome to raise moral issues. You can change like one base pair in the 6 billion base pairs and have a dramatic effect. And if that is extraordinarily expensive, you then do favor at least the rich government, the people of the rich governments, if they distribute their wealth throughout the nation. They won't necessarily distribute it throughout the world. So, for example, you can make a person resistant to HIV by messing up both copies of the CCR5 gene. You do not have to synthesize the genome to do that. And if you chose, you can make your whole population resistant to HIV. And you can make your whole population resistant to norovirus and a bunch of other things with very simple, currently available tools. And in fact, you can do it even without any synthesis at all by selecting embryos. So this is a profound question that we need to bring up again and again because it's already out there. Parents are already
stands between the Legion and annihilation.
Anecdotally, I have a 3-year-old boy, and he's got a younger brother expected in about 4 weeks. And the difference as a parent and another parent between the two of them is 3 years ago when Max was being born, we didn't know about his DNA. But Wiggles, as we call him, to be named still, has had his DNA sequenced in utero, where a blood sample from my wife is pulled and then using tools that George and others have made has been read out. And we are presented with that information and then basically have to make a go, no-go decision. So it's right to bring up the topic, but it's a topic that's standard of care in Palo Alto today. And it's a profound so, and I think it's, one of the puzzles is, if you're just presented with the thing that shows up, okay, I'll take what I get and I didn't have to. If I add intention to it, then it triggers a different type of reaction. Because now I have a type of responsibility, not only of choosing from the options presented, but the responsibility of choosing to take interventions up front in a different way. And I don't know that matters as much as I think George is right. The conversation's a live topic in response to your question, but not specific to synthesis, broader than that.
What is your Do you, like, there was a...
My
a
life
meeting,
is
I guess it was
dedicated
last,
to destroying
there
the
was an
burning
incident where people sort of worried that the Chinese
Don't
scientists
follow. I will
had
see to it that the Legion
fiddled
gets
around
no
with the human
farther.
genome, and there was a rumor
You,
that the
Corvus, and Sayana, attack the Legion from within. Then we'll wipe them out together.
Scientists said, oh, let's have a meeting, and let's just sort of worry together about, you know, and bring people from all over the world and say, here's some things we shouldn't do, and here's some, let's try to agree. So that was an attempt to self-regulate. Were you for that? Were you at it? And if you were at it, then how come you had a meeting like only like a couple of months later? Saying, oh, and let's now go do the human one. Because I don't, like, if it goes slow in this new project, they seem a little bit contrary.
So I was involved in David Baltimore's meeting and a paper that we wrote together. The meeting was at the National Academy of Sciences. And it wasn't really entirely self-regulating. There was numerous voices, probably not enough, different kinds of voices, but there was an attempt. There was a live stream for people could tweet in. But I think at the end of the day, it wasn't
How
that
can
different.
such a tiny
There was
thing
already
prevail against the
reluctance
might
to
of
do
the
anything
burning legion?
clinical with embryos before the meeting, and there was reluctance to do anything clinical with embryos at the end of the meeting. The research is still, legal in many countries and is being approved and extended, for example, in England. What the Chinese did was they were working with triploid embryos, meaning they had 3 copies of every chromosome rather than 2, and that is something that's not capable of developing into a full child. While there's work done in many laboratories before their work, working with pluripotent stem cells, which are capable of forming into every, at least in other animals that's been tested, into a fully developed mouse, let's say, in the case of mice.
Doesn't that raise this is the question, if you ever go to a Hollywood movie, the scientist is so often sort of unleashed. He's going to do something that scares all the other people in the movie. And that's not because Hollywood is just stupid. That's because Hollywood is smart. Hollywood has a sense of the science, and particularly the engineering culture, that it is so ravenous for newness and for development and for adventure and for knowledge that it scares other people because they are knowledge first, knowledge second, knowledge third. And when it's time for you guys to say no to something, I'm just curious, what would you now say no to, if anything, in this field?
Lots of things. I'd say no to making a virus with pandemic potential and releasing it into the environment. I'd say no to having...
That somebody actually did. Somebody actually made a polio virus, actually did it already, just published it even.
Yeah, and then what they showed was that from that capacity, they were able to do automated attenuation using computational algorithms to automatically generate the designs for a vaccine strain. So you could do rapid response vaccination if you needed to. By the way, let's keep going. Things I am against doing. The United States used to have an offensive biological weapons program. Matt Messelson and other scientists, using reasoning, were able to get the Nixon administration to shut it down. Let's keep, so there's a long history of things that we're for not doing. And I think it's healthy to raise the question. I'm a little bit, I think Hollywood's good at many things, including the making of money. And if I were to ask you, in the popular culture, what are the most famous movies offering perspectives on the future of biology and biotechnology? What might you remember?
I guess The Martian, Island of Dr. Moreau, Einstein, of course, classic, and Spielberg's Dinosaur.
What about Andromeda's?
Oh, I liked Andromeda.
And how about Gattaca?
Or didn't see Gattaca.
How about Contagion?
Where are you going with it?
So Contagion, Gwyneth doesn't come out too well in that movie. But the movie was released on the 10th anniversary to the day of the terrorist.
Save you. How can such a tiny thing prevail against the might of the Burning
The narratives, the
Legion?
telos, that give us not a naive nerd rapture sense of some future utopia, but this realistically plausible, hey, this is where we should aim. And you know, if you think about biology and you take a step back, we're not so egocentric to only talk about humans. If you think about biology as a type of material, living matter, super special, it's taken over our planet, how do we wish to partner with it? And what might our aspirations be? So let me just offer a few back-of-the-envelope numbers and then something that we could aspire to. So most of life on Earth is powered by photosynthesis. Light comes in, and it gets turned into chemical energy. And you can quantify how much energy that is. It's 90 terawatts of power on an average at any moment. Our civilization's running on of order only 20 terawatts of power. So deployed on our planet in nature is 90 terawatts in aggregate of photosynthesis. Human civilization's running on 20. Whoa. Maybe if we changed our interfaces a little bit between us and the rest of the planet, we could figure out how to flourish in partnership with nature. We could be transitioning from living on Earth to living with Earth. So, you know, somehow it's a challenge within the field, but I think it's a challenge that goes well beyond any of our scientific or engineering talents. It's the political and cultural and economic talents that are required here, the narrative talents. You know, we just have to get on a slightly different path. I don't want to industrialize biology. I'd like to biologize industry, and we should be able to pull it off, right? So what I've encountered over my life, born in 1970, human population has doubled. The ecologists are telling me the animal populations in nature have dropped in half. You know, I don't want to be part of that. And so how about we just aim this thing a little bit differently and partner with biology, living matter, to upgrade our interfaces, to not trash the planet, and not to be naive about it. But I think if you do the back-of-the-envelope calculations, we should be able to pull it off.
Let me ask you just one quick embarrassing question.
Advance! Destroy the Illidari! Take Azuna!
This is something that you decided not to do a couple of weeks ago. You decided not to go into a room at Harvard where there were. So we have some. Okay, so this is a meeting that was announced, and in this announcement of the meeting, and it's just a bunch of pros there. But if I could tell you in there, let me just see. It says something like, "We're not going to ask the press to come to this because it's closed by invitation." The phrase "closed by invitation" is in there, and then what happened next is Drew said he looked at this invitation and that this meeting was going on. This was a meeting to discuss making a human cell. And he said, as you can see there, he said, if you need to secrecy to discuss a proposed research project, then you're doing something wrong, he said. So you didn't want to go on. Can you just explain to me why didn't you want to, what was wrong with this meeting? I mean, George was in there. You in there?
Yeah.
I hosted it.
I couldn't make it. They scheduled it when I couldn't be there.
Okay.
Thank you for bringing it up. You know, as much as anything, an uncomfortable situation for me, and sort of feels like a fight among family. I don't really want to get into a discussion of making sausage. And I have to admit, folks who know me, but to say it out loud, I'm probably one of the most fanatical and excited proponents of doing these sorts of things. And my hope is, you know, I know we have another discussion, you know, later today on this whole
yes,
topic.
there'll be a second.
I don't want to unpack all of it, but I will share something, another sort of anecdote. My dad didn't go to college. He's got a wonderful set of siblings, 6 in total, and the youngest sister, my Aunt Baby, lives down in Cape May Courthouse, New Jersey, and she pays attention to what I do. And I was visiting one time for Thanksgiving, and at having a lovely family meal, and in the middle of the meal, she just blurts right out, goes right, "What's wrong with the DNA that God gave us?" and I hadn't expected the question, you know, nobody had given that to me in advance. And sort of after 20 seconds, I brought my jaw back up.
Because when lightning set fire to the chair next to you, you thought, oh, well, that's a good question.
Yeah. And I actually came up with a response I'm okay with. I said, "I don't know. Have you looked?" Not saying anything, have you looked? And that was really cool because
Have you looked at what?
Have you looked at the DNA that God has given you? I don't know. I'm not saying there's anything wrong with it, but have you looked? And she hadn't. And so that triggered sort of type of enablement. And within the next few years, she kept asking me, like, how do I look? How do I look? And I tried to help her. I could find some companies that were doing it. I wasn't totally comfortable with how they were feeding the information into various other companies. But finally, National Geographic, a student taught me about the National Geographic project to help people sort of understand their genetics.
We are the Legion!
And so my young boy, Max, when he was 2 years old, we sent Aunt Baby a kit. And then she figured out, she took a look. And it was so exciting, got a long letter back, and she was disappointed. She wanted to know more. She had learned that, she was from places she thought we should from historically, but apparently like 20% Japanese. Wasn't expecting that, and so on and so forth.
She had Japanese origin.
Not anybody knows about.
This is your aunt. That means you probably have some too.
Yeah. Who knows? I haven't looked yet. For me, I haven't looked. In any case, one of the cool things about the Moon program, when the United States, we sent a few people to the Moon, is everybody else pretty much had a chance to come along and experience it. Culturally, not physically. And so part of what I'm reflecting on is I love the specific projects that George and colleagues are proposing. But I also take at face value that this is going to go beyond that. And I can't wait. And I want to make sure that we're all going together. So I wouldn't make too much of family spat, so to speak. But you can if you want.
Well, I don't want to be like the reporter. I mean, I am the reporter. So what we do always is we just want to make sure we understand all the good things and all the not so good things about everything we look at. And sometimes people don't like us for doing that, but it is our job. So let me just come back to the question one last time, because we only have 13 seconds left on the clock, which is, would you guys mind if we spent like 10 more minutes here? You didn't want to go because this idea that if you need secrecy to discuss your proposed research, you're doing something wrong. That's the problem. So it should just turn off.
So I need to be careful, right? It's important for people to be able to meet in private. Yeah. And so there's nothing wrong. For me, when you're talking about developing a capacity that has the potential to be a general capacity to realize synthesis of human genomes, the human genome is something that connects us together. And so that's what triggered the response. Let me give you just the opposite end of the continuum. Under what conditions? You used some interesting words a while ago. You said doing such and such and such in humans, increasing virus resistance in humans. That implies actually doing it in humans, right?
In human cells.
Well, but you said you didn't say that.
Therapy.
Yeah.
Therapy is in humans, yeah.
But can I just say something out loud? Right? So it's like, could I imagine under any conditions where I would develop comfort and feel okay using such a capacity to synthesize the next generation of human beings? And under what conditions would I wish that possible? It's just my opinions, but when I reflect on that, I'll offer the two things that arise within me. Number one, I'd like to see culturally something akin to a sense of sacred in the realm of the digital, so that when I look at that sequence of the human genome in the computer or whatever the holographic interface is, I feel the same way about that information as I do about my boy. All right? So if that were true, that'd be a type of condition I'd want to work through. Second condition, I'd like to see humanity and nature flourishing together. I think it's a little bit of a distraction to focus only on people. And I'm also going to respect that there's going to be a plurality of positions, that are going to be opposition and so on. But it's something I can imagine. And because I can imagine it, and because I believe it's totally doable, right, that it's worth having a variety of conversations.
I suppose, without putting words into your mouth, that you thought, well, we have an article going into a magazine. There's a period where you're supposed to be quiet before the article is published. So you know how reporters are. So let's not have them in there. And maybe this was sort of a mistake, but because you tell a reporter to stay out, then they're going to pretend they're a lampshade.
Too late, heroes. Your
I
world
don't
is
recall
lost.
asking reporters to say, I think we had 3 journalists present. I think, and I don't think that the embargo of the paper was a major motivator from my standpoint.
Okay.
As host, I was trying, I think, respect. I agree with Drew that one of the cool things about the Apollo program is we were on board. But we weren't present at every single meeting leading up to the Mercury and the Gemini and the Apollo. There were some meetings that were not public. And I felt that there were people in the room, many people in the room, who wanted to talk about things which were not yet published, that they had not yet even framed how they wanted to present it to the public. There were preliminary thoughts that they thought could get distorted and that they didn't get the words right. And they wanted to develop those words in a small meeting, relatively small meeting, it doesn't matter what the size is. And I wanted to respect those wishes of those people. In fact, we intended from the beginning to make quite a bit public, including videos of the conference, which is bringing people into the space capsule in a certain sense. But there are some people who still do not want their particular video to be public. And I respect that. I do not want to rush them, because if we rush them, then they might get their message garbled, as exactly what has happened, because they have been rushed.
Let me ask you one last question. I noticed that the paper that proposes all this has just been published, and the title, I'll show you the title of the blog, is called The Genome Project - Right. In the earlier draft, which was in that meeting, it was called The Human Genome Synthesis Project. So somehow human was dropped, so now the title is just the Genome Project. Is that because you thought, let's just not scare everybody, or is that because you actually changed your mind about what you wanted to do?
So I can share my personal vendetta against the word the Human Genome Project. I was involved in it from before it existed. 1984, I came up with it in a meeting where we didn't know that's what the meeting was about, but turned out that's what it was about. And I was opposed at that point to saying we were going to sequence the human genome, because I thought there were so many other genomes. And in fact, it would be meaningless about comparisons. If we have a comparison between human and chimp, and human and mouse and chimp, and lots of comparisons, we would learn much more. So I felt it was inappropriate, but I was outvoted. Of course, I was the youngest member at the time. This time I was still outvoted, even though I was the oldest member of the committee. This is tough.
Tall is not good enough, apparently.
But I think that the nice thing about these conversations is it is a plurality, and I made no effort to talk over the people in the room or the people outside the room. I've been on papers that have had more authors than that paper that just came out, or at least had more signatories on it. I've never been in one where there were more people micromanaging the paper than this one. This has a real record for the number of people engaged, people who are not authors, people who are not editors. Everybody was putting in their 2 bits, and so that is a work of committee effort. But I will take full credit for trying to get rid of the word human from it. Not because I'm not interested in human. Probably of all the people that signed that, we have the most active research on engineering human cells. And actually even engineering human beings. I have to take responsibility for that too. Because one of my companies, Editas, is developing gene therapies using CRISPR. That's engineering human beings. And there are 2,000 gene therapies in clinical trials already.
I guess I'm being told to shut up on either side of the stage. So, because here's what I was going to do at the very end. So I guess I'll just do it, and then all I'll just do is make you raise your hands or not raise your hands because it's not fair. But I was going to end and ask you all four of you, will we be able to make people with artificial genomes? And if we are one day able to make people with artificial genomes, should we? That doesn't actually get asked at the end of the day. So it's 0, 0, 0, so I can't. I'm just curious. Would any of you raise your hand and say yes? One day, if we can, we should, or shall we?
We're already doing
all
it.
four.
Maybe we should ask the audience.
Yeah. Let's just see. Show of hands. If you could,
Look at
would
that.
you? All right. Hands down for you. And if you could, shouldn't you? Let's not.
Wow, that's interesting.
Only a very few.
No.
You see, that's why they put
With
the modern...
FDA approval.
Thank you very, very much for showing up. Thank you for the extension. Thank you, Al. Thank you, Science Festival. Thank you all.
Do not give in. We will work together to destroy the Legion from within.
When you spend all year serving others, you don't just land on the nice list. You get rewarded by becoming the top brand in new vehicle quality in the auto industry. Celebrate with a lineup of Ram trucks built to serve.
My life is dedicated to destroying the Burning Legion.
The rules of our universe seem surely seen. This solid bridge supports my way. The water flowing underneath always goes downhill. And
Remember,
when I throw this
nothing is
stone,
more important than the Legion's
it always
destruction.
flies through the air following a predictable path. But as scientists peer deep into the tiny building blocks of matter, all such certainty vanished. They found the weird world of quantum mechanics. Deep down inside everything we see around us, we found a universe completely unlike our own. To paraphrase one of the founders of quantum mechanics, everything we call real is made up of things that cannot be themselves without. Around 100 years ago, some of the world's greatest scientists began a journey down the rabbit hole into the strange and the bizarre. They found that in the realm of the very small, things could be in two places at once. That their fates are dictated by chance. And that reality itself defies all common sense. And that everything we thought we knew about the world might turn out to be completely wrong. The story of our descent into scientific madness begins
I don't have a target.
Germany is a new country, recently unified and hungry to industrialize. In this newly unified Germany, a number of new engineering companies were founded. They have spent millions buying the European patent for Edison's new invention, the light bulb. The light bulb was the epitome of modern technology, a great optimistic symbol of progress. engineering companies quickly realized they were fortunate. Strangely enough, this humble object is responsible for the birth of the most important theory in the whole of science, quantum mechanics, a theory that I've spent my life studying.
We are all that stands between the Legion and annihilation.
Illidari, what happened here?
Engineers knew that if
I didn't give in.
Electricity, it glowed. The physics that underpinned this, though, was completely unknown. But something as basic as the relationship between the temperature of the filament and the color of light it produces. In 1887, the German government invested
It's
millions
too
in
far
a new
away.
technical research institute here in Berlin. The
I need to get closer.
Then straight-laced scientist to help work here. His name, Max Planck. Planck took on a deceptively simple problem. Why the color of the light changes as the filament Forget about the bubble taking the light. With an old-fashioned lamp powered by
My life is dedicated to destroying the They have all been tortured.
They did things the brighter the light. The more I pedal, the more electricity the dynamo produces, the hotter the filaments in the lamp, and the brighter the light. But the light the bulb makes isn't just getting brighter. It's changing color, too. As I speed up, the color shifts from red to orange to yellow. Right, now I'm going to really belt it. Now the bulb's filament is getting even hotter, but although it certainly gets brighter, the color seems to stay the same, yellow-white. Why doesn't the light get any bluer? To investigate, Planck and his colleagues built this, a black body radiator. It's a special tube they could heat to a very precise temperature and a way to measure the color or frequency of the light it produced.
What did they do? What did the demons offer you?
The demons promised us unimaginable power.
Light it produced.
You're not supposed to be here!
Nowadays, over 100 years later, the people are still doing exactly this kind of measurement, just much more accurate. The temperature inside here is 840 degrees centigrade. Feel the heat coming off, and it's glowing with a lovely orangey red colour. It's about the same colour as my bike light when I'm cycling slowly. But I want to see something hotter still.
I'm out of range.
Here is about 2,000 degrees. Light. To produce light of this intensity and colour requires a power of about 40 kilowatts. Now, that's equivalent to...
I need to get closer.
I'm fucking very fast. or the combined...
It's too far away.
Even the sun, at a temperature of 5,500 degrees centigrade, produces mostly white, visible light, and makes remarkably little ultraviolet light, given how hot it is. Why is this? Why is ultraviolet light so hard to make? This remarkable failure of common sense so perplexed scientists of the late 19th century that they gave it a very dramatic name. They called it the ultraviolet catastrophe. Planck took a crucial first step to solving this. He found the precise mathematical link between the color of light, its frequency, and its energy. But he didn't understand the connection. However, it was another weird anomaly that would really put the cat amongst the pigeons. In the late 19th century, scientists were studying the then newly discovered radio waves.
Your life ends now!
To do that, they were building jump between the two metal spheres.
I need to target something first.
Something very unexpected to do with light. They found that by shining a powerful light source on the spheres, they could make the sparks jump across more easily. This suggested a mysterious and unexplained connection between light. Scientists used it. It's called a gold leaf electroscope. It's basically a more sensitive version of the spark apparatus.
Who did this to you?
First of all, I have to charge it up.
The warden tried to convince us to turn traitor.
What I'm doing is adding a set of electrons that are pushing the two gates.
My eyes see all.
Red light. Immediately, gold leaf.
Did anyone give in?
Cyana Nightglaive turned traitor. She joined the Legion.
Light can clearly remove static electric charge from the leaves. It can somehow knock out the electrons I've added to them. But why is ultraviolet light so much better at doing this than red light? This new puzzle became known as the photoelectric effect. The ultraviolet catastrophe and the photoelectric effect were big problems.
I'm out of range.
Because neither could be understood using the best science at the time. The science that said quite unequivocally
Medium or well done!
I don't have a target.
Shadow might be fuzzy around the edges. We understand this as the light bending, probably common sense wave-like behavior. And here's something else.
I need to get closer.
Look at these soap bubbles. Shine a light on them, and gorgeous colored patterns emerge from nowhere. And this was easily explained if you accept that light was a wave, reflecting
You cannot hide from me.
Thin soap film. And this was a problem was this: how could light do this? To truly grasp how absurd this phenomenon was, it might be useful to consider how waves in water
Lord Illidan knows the way.
It's used to train lifeboat teams to deal with a range of different kinds of water waves. First, small waves, just 30 centimeters.
My life is dedicated to destroying the burning legion.
Where did Cyan and Nightblade go? I need to find her.
Nightblade went southwest with a warden named Cordano Felso.
A warden working for the Legion? I look forward to killing her. I've sacrificed everything. What have you done?
It's clear what water waves are telling us. Bigger, more intense waves have more power. They easily knock me and the cans around. So if light was a wave, more intensity should knock out more secrets before you die. That's not what
I need to target something first.
Thinking of light as a wave just wasn't adding up. To resolve this, someone needed to think the unthinkable. And in 1905, someone did. You may well have heard of him. His name was Albert Einstein. Perched on top is a strange, huge iron and steel construction. But it's not a gun. It's actually a telescope. Built in 1896, the telescope was one of the largest of its kind in the world and made the observatory the go-to place to engage and astound the public in new science. Albert Einstein gave a very famous public lecture here on his theory of relativity, which is, of course, what he's most famous for. But it's not the work that won him the Nobel Prize. To Einstein, a quantum was a tiny number of energy. And although in 1905 the word wasn't new, the idea that light could be a quantum seemed crazy. And yet, following Einstein's heretical line of thought to its logical conclusion, solved all the problems with light at a single stroke. I'll try to explain how this helps using a rough analogy. Of course, like all analogies, it's far from perfect, but hopefully it'll give you a sense of the physics to help you understand why thinking of light as a stream of particles solves the mystery of the photoelectric effect. In this analogy, these red balls represent Einstein's light quanta. And those cans over there are the electricity held in the metal. Now, in the original experiment, they made electricity flow from the surface of the metal by shining light on it. In my analogy, I'm going to try and knock those cans over using these red balls. absolutely no effect. That's just like red light.
Quickly, we have to find the key to those demonic
Each
locks.
particle of red light
I'd bet anything
carries
it's on that
very
big
little
jailer
energy,
to the east.
because red light has a low frequency. So even a very bright red light with many red light particles can't dislodge any electrons from the metal plate, just like the red balls. Now I'm going to use heavier balls, like these blue golf balls, and I'm going to try and knock off the tin cans with these. Just a few dim
Get the key. Let's get over there and free that Drake.
Being blown away by sheer elegance and simplicity. But what's more, Einstein's nifty idea
You will soon be
also
free,
helped
Drake.
solve Frank's mystery of the light bulb. It's more red than ultraviolet, because ultraviolet took so much more that's 100 times more energy. No wonder there are so few of them.
That moment at the beginning of the 20th century signaled a genuine revolution because it demonstrated that the claim of physical science people were doing right back to Newton and like that you needed a
Thank
completely
you.
new approach.
I am Stella Gosa.
Physics has never recovered from that moment in the sense it's built on that moment. That's where the really
Range.
Also left physicists with a grisly problems to find all common sense. Light was definitely a wave, which explained shadows and bubbles. And now it was definitely a particle too. Einstein's concept explaining the photoelectric effect. Einstein's brilliant, crazy idea. Because what seemed to be a curious mystery about light was about to become a battleground like the nature of light.
My thanks. Now I am going to finish off the warden who did this to me.
Stella Gosa, no! Come back! Damn!
1992. The Western world was in the grip of James Joyce's Ulysses is published. Stravinsky is at the height of his powers, and Chaplin has just released his first series of movies. The Ottoman Empire collapses. Europe is still recovering from the war to end all wars in which millions of men lost their lives. Russia is newly communist. Meanwhile, America is exporting jazz to the world.
Lord Illidan knows the way.
In art, politics, literature, economics, there was an insatiable appetite for change. This was the birth of modernism.
I have to find Stella Gosa and Cyana Nightglaive. Meet me up at the Overlook. We will finish this business together.
Come, do your best. Nightglaive has been undergoing some changes.
But, and
We are
I might get
ready
in trouble
for
for
you.
saying this, I would argue that the upheaval that took place in physics at this time would eclipse them all and have far longer lasting consequences. It had begun with the discovery of the weird and contradictory wave-particle nature of light. It ended up as an epic battle fought between the greatest minds in science for the highest possible stakes, the nature of reality itself. revolutionary scientists and their leader, the brilliant Danish physicist Niels Bohr. On the other side, the voice of reason, Albert Einstein, at the height of his powers, now world famous, a formidable adversary. The battle raged for decades. Actually, in some ways, it still does. It was fought across the world in universities, at conferences, in bars and cafes. It would reduce grown men to tears. And it began with a deceptively simple experiment. But weirdly, it was an experiment that wasn't even about light. It was about the particles that make electricity. In the mid-1920s, an experiment was carried out at Bell Laboratories in New Jersey in America, which uncovered something entirely unexpected about electrons. Now, at the time, it was accepted without question that electrons were these tiny lumps of matter, small but solid particles like miniature billiard balls. In the experiment, they fired a beam of electrons at a crystal and watched how they scattered. Now, that's entirely equivalent to taking a beam of electrons, say, from an electron gun, and firing it at a screen with two slits in it, so that the electrons pass through the slits and hit another screen at the back. What the Bell scientists found shocked the physics world to the core. To understand why, consider a similar experiment with water waves. I set up a simple experiment. I have a water ripple tank placed on top of an overhead projector. I have a generator producing waves that pass through two narrow gaps. The projector beams the image of the waves onto the back wall. You can see as the waves come in from the left and squeeze through the two gaps, they spread out on the other side and interfere with each other. What this means is that when you get the crest from one wave meeting the crest from another, they add up to make a higher wave. But when the crest from one meets a trough, they cancel out. This gives rise to these characteristic lines leading to the signature wave pattern. Bands of light and dark. Whenever you see these light and dark bands, the signature wave pattern, you know without doubt that you've got
Fellsworn
wave behavior.
Nightglaive, prove yourself. I have other matters to attend.
Yes, my mistress.
Do not give in!
I am more powerful than all of you!
And now, electrons for a long time
Only knew the Legion's power. Join us, Corvus.
Never! You and your new mistress.
I need a target.
But gradually a pattern forms.
Let's get out of here before we're overrun.
Let me be quite clear about just how weird this is. Remember from the wave tank experiment where the signature wave pattern only exists because each wave passes through both slits and then it But here, every individual electron, each single particle, is passing alone through the slits before it hits the screen. And yet, each single electron is still contributing to the signature wave pattern. Each electron has to be behaving like a wave.
No one can be allowed to become a fell-sworn traitor working for the Legion.
This strange result, Niels Bohr and his colleagues...
We've dealt them a telling blow.
Quantum mechanics.
But even now, the Legion is summoning in more powerful demons.
That embraced contradiction and didn't care that it was almost impossible to understand.
We cannot allow them to regain their momentum.
Anyone who
The broken
isn't shocked
isles
by
are
quantum
lost.
theory hasn't understood it. So, viewers, I'm going to take
You
our
must come
tiny
to the
electron
Azure Wing
and use it to
repose
delve
and
deep
meet
into the
with my
heart of reality.
grandfather Senegos.
Your Titan gods have abandoned you. Lay down your arms. There is no hope left.
Quantum mechanics says this. We can't describe what's traveling as a physical object. All we can talk about are the chances of where the electron might be.
Azeroth must not
This
fall.
wave of chance somehow travels through both slits, producing interference just like the water wave. Then, when it hits the screen, what was just the ghostly possibility of an electron mysteriously becomes real. Let me try and capture just how weird this is with an analogy. If I spin this coin, then all the time it's spinning, it's a blur. I can't tell if it's heads or tails. But if I stop it, I force it to decide, and it's heads. So before, it was sort of not heads or tails, but a mixture of both. As soon as I've stopped it, I've forced it to make up its mind. This is what Bohr and his supporters claimed was happening with our electrons. In a sense, as it spins, the coin is both heads and tails. Similarly, the electron's wave of chance passes through both slits, two paths at the same time. Our coin then stops at heads. The ethereal wave of probability hits the screen.
Knowledge is power, but using it wisely is the key.
The quantum world was unlike anything ever seen before. It's hard to overstate just how crazy this is. Bohr
Do
was
not
effectively
linger here too
claiming
long.
that
The
we
Legion is
never know
attacking
where the
everywhere.
electron actually is at all.
I am certain that if anyone can help you find this pillar of creation, it will be my grandfather.
Weirdly, as though the electron itself is everywhere.
I'll meet you up there. You will find repose up the road to the northeast.
Electrons are among the commonest and most basic building blocks of reality. And yet, here's Bohr saying that only by looking do we actually conjure their position into existence. It's like there's a curtain between us and the quantum world, and behind it, there is no solid reality, just the potential for reality. Things only become real when we pull back the curtain and look. And this view, ladies and gentlemen, became known as the Copenhagen
That weapon will not save you. How can such a tiny thing prevail against the might of the Burning Legion?
And they found a natural leader in the most powerful man in science. Albert Einstein hated this interpretation with every fiber of his being. He famously said, "Does the moon cease to exist when I don't look at it?" He was very unhappy because it gave limits to knowledge that he didn't think should be final. He thought there should be a better underlying theory. Over the next 10 years, Einstein and Bohr would argue passionately about whether quantum mechanics meant giving up on reality or not. Then, with two other scientists, Nathan Rosen and Boris Podolsky, Einstein thought they'd found a way to win the argument. He was convinced he'd found a fatal flaw in the Copenhagen interpretation, and its claim that reality was summoned into existence by the act of looking at it. At the heart of Einstein's argument was an aspect of quantum mechanics called entanglements. Now, entanglement is this special, incredibly close relationship between a pair of quantum particles whose fates are intertwined.
This does not look
if they were
good.
created in the same
He
event.
is not well. What is happening here?
Let the useful
Let me try
one
and explain this by
approach.
imagining the two particles
I am
spinning
not
coins.
as dead as I look.
Imagine these coins are two electrons, created from the same event, and then moved apart from each other. Quantum mechanics says that because they're created together, they're entangled. And now many of their properties are forever linked, wherever they are. Remember, the Copenhagen interpretation says that until you measure one of the coins, neither of them is heads or tails. In fact, heads and tails don't even exist. And here's where entanglement makes this weird situation even weirder. When we stop the first coin and it becomes heads, because the coins are linked through entanglement, the second coin will simultaneously become tails. And here's the crucial thing. I can't predict what the outcome of my measurement will be, only they will always be opposite. Einstein seized on this because it meant that something was happening between the two coins that was almost too crazy to imagine. It's as if the two coins are secretly communicating instantaneously across space and time, even if the first coin was on Earth and the other was on Pluto. Einstein refused to believe this instantaneous, faster-than-light communication. His theory of relativity said that nothing could travel that fast, not even information. So how could one coin instantaneously know how the other would land? He disparaged.
Yeah, tune in next time when I will be available again to take your calls. For some reason, you just don't want to call. Oh well. I'll be here though. Don't you worry about it. I will be here until I die. And so long.